Hello, everyone. Welcome to the lecture on biofuels. Okay, this week's lab activity is going to be on developing biofuel in the teaching lab and getting ethanol from various sources. So some background, right now ethanol fuel is experiencing a period of growth. In the US, it's generally added to fossil fuels or to gasoline in the form of blends, which meet the octane requirements. But in Brazil, they're actually providing a successful model for the use of ethanol fuel as a single fuel, not as a blend. And after 30 years, Brazil has nearly replaced fossil fuel with ethanol, and they produce it from sugarcane. So for example, there's a Fiat model, I forget the number, that runs entirely on ethanol fuel, and it's used in Brazil. So in the US, growing sugarcane is impractical, and instead corn is the primary carbohydrate source that's used for ethanol for fuel. Here's a picture of an industrial ethanol plant. They produce ethanol through the fermentation of domestically grown corn. And these giant columns are actually giant fractionating columns for purifying the ethanol from the fermentation broth. So they're basically doing a fractional distillation, but on giant scale. So there are some objections to corn-based ethanol. They are both ethical and practical. Some people object to ethanol being used for fuel because it diverts land away from food crops. So if you're growing corn for the purpose of producing ethanol, then that land is not being used to produce food crops. Corn does not produce as much sugar compared to sugarcane. So it's a question of, is the land that we would need to devote to corn production, is it an economical and smart way to produce ethanol for fuel? The production of corn, or the growing corn for ethanol, has significant material costs and energy input. So it requires you to put in energy in order to get the corn that can be converted to ethanol that you can use as energy in a fuel. And the energy input, for example, is the energy needed for plowing, seeding, irrigation, harvesting, and the application of fertilizers and pesticides. So all of that has to be done, and it takes energy to do that. And it's the question of how much are you getting back for your energy input. So there are some alternatives to corn that are still being developed to this day. So people are studying them in academic labs. One alternative is using perennial, meaning you can grow it year long. Cellulosic sources such as grass. They offer promise because large quantities of grass can be harvested with very little energy input for upkeep. So you don't have to use fertilizers, pesticides, you don't have to water it a lot, right? So the amount of energy needed is pretty minimal. So that's why it's being looked into, although there are some negatives that we'll talk about as well. The other option is waste from food crops. So waste biomass such as wheat straw and corn stalks. Those are promising because we're already growing corn and wheat, right? So you don't have any additional cost or energy input because those materials are waste and they're already being grown for food. So this leads us into the idea of switchgrass as a particular type of grass that's very promising. So I mentioned in the last slide that there are some problems with using grass as a source of glucose, which can be used for the fermentation to produce ethanol. The problem is that the grass, the carbohydrate source in grass is cellulose, and that's a little bit harder to break down into glucose. We'll talk about that as we move through the slides as well. And the major problem, however, is the molecule lignin. It's a structural component of plants, and it's a very large hydrophobic molecule. You can see the structure on the slide here, and it's not easily separated from cellulose. So that's a bit of a problem because this is not a glucose source, right? Lignin is not, but it's very difficult to separate from the cellulose that is then the carbohydrate source that produces the glucose. So the last issue of lignin is why research laboratories are exploring alternatives to corn. And a lot of the focus is on switchgrass because it does not contain lignin, which solves one of the major problems of using grass as a source of ethanol. Okay, this particular experiment looks at isolating ethanol from 5 different sources of glucose, right? And those are, we're going to use apple juice, which obviously comes from apples, grape juice, which comes from grape, potato starch, frozen corn, and then when the students do this in the lab, typically we don't use grass. The data is just provided for grass. But I'm going to talk about why and how you would use grass if we were doing an extension of the experiment that included grass. Okay, so we are looking at alternatives to corn and including corn in our analysis to produce ethanol, right? So it's a very interesting because a lot of times people, they know that ethanol comes, which is used for drinking alcohol, right, is produced through the fermentation of sugars from barley and certain grain sources, right, to produce alcohol, drinking alcohol. But they don't typically think about ethanol's use as a biofuel. So that's kind of an extension of that we can really utilize in the lab as an interesting and practical application of organic chemistry. Okay, so we are going to look at a bunch of different carbohydrate sources. We're going to calculate how much ethanol can be produced from them and do some interesting calculations. Okay, let's talk about fermentation. Fermentation is an anaerobic process, so we want to exclude oxygen from the reaction mixture. And it's a process where yeast converts glucose into ethanol and carbon dioxide. So we mentioned that we're going to be using 4 different sources, right? And this is a round bottom flask being used for the fermentation of one of the juices, okay? And so the fermentation, what does it involve, right? So the carbohydrate source, which could be a fruit juice, again, it could be potato starch or corn, although potato starch and corn require a pre-treatment prior to fermentation that we're going to talk about. But once the carbohydrate source is ready, it's placed in a round bottom flask with a glass adapter connected to some Teflon tubing. A glass pipette is inserted into the tubing on the end, which is down here, right? And then it's submerged below the surface of saturated aqueous calcium hydroxide. So that's the lime water that's mentioned in the picture, right? The purpose of this is to exclude oxygen from the atmosphere from getting into the reaction, and also for a place for the carbon dioxide that's produced in the fermentation to bubble out and collect. So that's the purpose of that lime trap. Anyway, the fruit juice, let's look at what needs to be done to the fruit juice in terms of a pre-treatment. Actually, nothing needs to be done to it. Place the juice directly with the yeast into the fermentation flask. 100 milliliters of the juice in both cases are used, and we don't need any additional nutrients or changes in pH to grow the yeast cells. Everything that is needed is already there. But I mentioned that both the corn and the potato starch require a pre-treatment, because with fruit juice, most of the carbohydrate source is already glucose or a dimer, such as fructose, which can be converted easily by the yeast into ethanol. But the primary carbohydrate source in corn and potato starch is starch, right? And that starch needs to be broken down into glucose prior to setting up the fermentation with the yeast. So for corn, 100 grams of frozen corn would be pureed with 50 milliliters of water in the lab. And if a student group had potato starch, they would take 50 grams of the potato starch and mix it with 100 milliliters of water. Then they would take a concentrated acid solution, specifically 25 milliliters of 6 molar HCl, add that to the corn or the potato starch solution. The mixture is then heated on a hot plate to 90 degrees for 45 minutes. And the purpose of the acid is to hydrolyze the polysaccharide in starch down to smaller pieces, which are primarily going to be, hopefully, be glucose at the end of the acid treatment. If you didn't want to use acid, alternatively, you could use amylase. Amylase is an enzyme that can break down starch into glucose. At the end of that acid treatment, the mixture is neutralized with base, and the pH is adjusted to approximately 6.2 with a phosphate buffer, and students would double check with pH paper. Then they'll want to double check the glucose content to make sure that it's sufficient to move on to the fermentation process. So there are glucose test strips made by Carolina Laboratories, and they want to make sure that the concentration of glucose is at least 0.1 molar. If it wasn't, they would have to add acid again and heat again, which would take more time. So the idea is hopefully they did a good job in the first place, and there's a high enough glucose concentration to move on. At that point, 3 grams of yeast is added, and the fermentation is carried out as described on the previous slide. So we, in the lab, we do not do the grass, and you're going to see why as we go through the procedure for breaking down the cellulose in grass. So if you wanted to do it, you would take dry ground up grass, about 2 grams. It's pre-treated by heating it to 90 degrees in a basic environment for 90 minutes and then washed with water. The point of this is to help remove a lot of the other cellular components in grass so that you're mainly left with the cellulose that you want to move on with. At that point, the treated grass is stored in a refrigerator until the day of the experiment. The treated grass is then incubated with cellulase enzyme. So notice that we're not talking about the same enzyme we were using for starch, right? Amylase enzyme is used to break down starch into glucose, but if you're breaking down cellulose, you need the cellulase enzyme. So you need to incubate it with the enzyme that can break down cellulose. And because enzymes are particular about their environment, you need to use a buffer at a particular pH, and even add some ampicillin, which is an antibiotic, to prevent bacterial growth. And you would have to do that for 24 hours. So the fact that that needs to be done for 24 hours prevents us from easily doing it in the lab because of the time constraints of 3-hour labs. So the idea is that the enzyme would break down the cellulose into glucose, We would measure the glucose concentration, just like we did before, make sure that it's sufficient concentration and that we have the right pH for the yeast to be happy. So the yeast is then added and the fermentation would be carried out at that point. Let's talk a little bit about the structural difference of starch versus cellulose. You'll notice the region that is highlighted, which is the linkage portion of the individual glucose monomers that make up the polymer of starch and cellulose. The linkages are actually different, specifically the stereochemistry about this carbon, which is known as the alpha carbon to this oxygen, right? The stereochemistry is different. You can see here that carbon is the hydrogen bonded to the carbon is up, the oxygen is down, right? Versus cellulose has the oxygen up and the hydrogen down. So the RNS configuration about this carbon would be the opposite. And that is the difference between the two polymers of glucose. Of course, acid could break down either linkage, but if you're doing it enzymatically, you need an entirely different enzyme to break that down. Humans contain a lot of the amylase enzyme. So the breakdown of starch begins in the mouth, right, and continues through the digestive system. But humans don't contain the cellulase enzyme, so we cannot break down things like grass. But other animals, like cows, definitely do have it and can break it down. So those linkages are called alpha 1,4 and beta 1,4, referring to the direction of the oxygen. Okay, so the fermentation is allowed to go for an entire week, but at some point the alcohol or ethanol production will stop. And that happens at a maximum concentration of ethanol in the fermentation broth of around 15%. Most yeast strains will die once the ethanol concentration gets up to around 10 to 15%. So it will stop at that point. Once the yeast is dead, no more fermentation can occur. After a week, students will filter the mixture, which will remove any particulates such as yeast and other particulates that might be in there, which is like with corn, there's a lot of particulates in there to filter off. And then the liquid that results from that is called the fermentation broth. The students measure the volume of the fermentation broth that they got, but they only take 100 milliliters and move on to do the fractional distillation with that volume, leaving the rest to the side. Okay, so from here, I really want to talk a lot about the fractional distillation process, the theory behind how miscible liquids are mixed and how they're separated through fractional distillation. And so I have an entirely different PowerPoint presentation devoted to the theory behind that and talking about the fractional distillation that the students do on the ethanol water mixture that is contained in the fermentation broth. So stay tuned for the next video.